docking. I had done, a lot of training. I have seen people misuse it. That's why, as we always say, you all are, uh, most of you are like next generation, young people with a lot of dreams and all. But you all know that always we should have a good idea about ourselves, and we should be our best judge about what we should try, what we are good at. Other people could not. So in our desi language, we call about okat. We should know about what is our okat. And when it comes to molecular docking, I don't know why people do like lot of works which are actually not possible. People give importance to things which in theory clearly written that it should not be given that importance. Uh, so my note is, whenever you learn docking, whenever you want to apply docking in future, remember this slide. And with that, I would ask people to be very understanding, because a lot of people are coming in the platform. So please mute your microphones unless it is required. Okay. So when we look at docking, you should get the memory of the childhood game of matching you used to play, because docking attempts to find the best matching between two molecules, between a macromolecule and a small molecule, and not more than that. What I'm trying to say, when we consider biology, the biology of a protein is not only completed with the 3D structure of the molecule. There are certain flexibilities, there are certain change of conformations which are not estimated by this matching, which is coming through the algorithm of docking. So in theory, docking would be between protein, DNA, and all these things. But in reality, docking match only in case of protein and small molecule. And that also should be carefully backed up by a lot of literature. I have like seen a lot of work coming, some are somehow published, where people are taking COVID-19 targets like protease, like RNA polymerase, like spike protein, and did a lot of work, did a lot of work. Let's say I have seen, and you all know that 2DG is a molecule which was innovated by DRDO scientists and coming into market. And a lot of people are taking 2DG and making docking studies with any and everything, like with protease, with the spike protein, with uh, RNA polymerase. You have to understand the fact that when a drug is coming, if you are interested with that drug, go and read, know about the chemistry, know about the target they are mentioning. Because if you are picking, and some of you might argue, if we have a wrong target, the docking interaction should not be provided well. No, that's not true. Because protein is a macromolecule, it have a lot of bigger size, bigger 3D scaffold. There might be a corner where you have some affinity for the compound, it is going there, docking there, and you get the data. What I mean by that, you learn docking. Docking is a good tool. Docking is very good tool to work to know about protein drug interaction, it is very useful tool for going to virtual high throughput screening, by which we could replace the high throughput screening, and that is instrumental in reducing time, reducing money. But before going into the docking tutorial, promise me that you would not do docking by keeping your brain elsewhere. You would not believe only the technical part of the docking, but whenever you are doing docking, you would parallelly use your brain to what is happening. If you are in Calcutta and if the storm is not there and you still model it, what would be the effect, and put other cities effect, and so theoretically a lot of destructive effect, it could not be happening because the storm is not coming there. So that much gaps you would be taken care. Please promise me. And with that, I hand over it quickly to Vivek, because as I told, I had an idea to give you much bigger introduction, which I'll come in the next week. But this week I allow Vivek to continue quickly, so that how much power backup he could have finished this. So Vivek,
Yes, sir.
yeah, I'm just removing my slides and then it is your turn.
Yes, sir.
I have done that. Okay.
Okay.
Again, thank you everyone for coming. Let's Vivek start the journey. Thank you. But again, before I finish, remember, docking is not God. Docking is not the solution of all of your problem. Docking is an important tool for drug designing, drug development. But if docking is the technique, your brain is what you need. Thank you.
Okay. Good morning to all. So, uh, is my screen visible?
Yes, we can see.
Yes.
Yes,
Yeah.
yes.
And somebody has started the recording. Is it going on? Matlab, somebody has started it, right?
Yes, it is
Yes, sir,
going.
it's running.
Yeah. Okay. So let's start.
Vivek,
So
sorry.
yes, sir.
You always start recording from the time it's on.
Yes,
We
it is
need,
already started from that
yeah,
point only.
yeah,
I
yeah.
just wanted to confirm. Yeah.
Yeah, someone say they cannot hear. So take care. Ankika Vak is saying that she cannot hear.
Okay.
Yeah.
It's still the same. Matlab, sir, you can hear me well, right?
Yeah, I can hear you. I can hear you.
Yeah. So maybe
I
the
can hear, I
internet
can see
issue.
the slides,
Yeah.
everything is working perfect, and just continue. Okay.
Okay. Okay. So I will start with the prerequisites for our docking. So first is the protein target, which is has to be in the .pdb format. The small molecule structure to which we are going to dock. Literature study of small molecules towards its activity as a substrate or cofactor or inhibitor, what it is, and detailed study about the active site of the residues of a protein, and study important loop and their effects. So these are the prerequisites. I will talk about each and everything in my slides. So due to as sir told, due to the present circumstances, I will not able to provide you hands-on training, like exactly showing it, because if I open the apps, it will reduce my power backup. So that we will perform, the tutorial we will perform in the next class, where I will tell you what to do and you will do the exact each and every step. But today I will go through those steps and I will tell you. Meanwhile, you can also try and we can discuss your problems too. So first, what we are discussing today is we are discussing the docking of small molecules, drug molecules, to the protein or enzyme target. Know that this is just a drug and a protein. There is no docking. We are discussing today about protein docking or any protein RNA docking. We are just discussing about protein and small molecule docking. Okay, so for a docking, what you need first, you need enzyme structure. So in the previous our previous tutorial, we talked about homology modeling and how we can get the structure if it is not experimentally solved. Okay, and how also we can get the structure of a US sequence from RCSB database. So that we have covered. So now what we need is a small molecule structure, right? You have a drug, but you know from where you will get the structure of it. So first thing is, for that you need, you have a different databases. The I have enlisted few of them. There are more also, but you can go through them and you can see the get the structures of this small molecules, because sometimes you will not be having structures, like you will not having a 2D structure. Okay, some of them gives you 3D structure as well. So these are few databases through which you can get the structure of small molecules. But uh, what are the problem with those? Okay, but suppose you have a new structure which is not reposited anywhere. So what you will do? What are the choices there? Okay, so know that whenever you get any structure from these databases, you will perform the few steps which I am going to discuss with you, like energy minimization. Also, you have to do on the structure which you are going get through such a databases. Okay, so you will get the structure in mainly mol and SDF format. If the database gives you in PDF format, that is also well good, but you have to convert them all them into the .pdb format. Okay, so this is the one. The first one I'm showing is ChemDraw. Okay, so using the ChemDraw, you can see, I guess, all the, like, there are benzene, 5-membered ring, 6-membered ring, 4-membered ring, the sticks, double bond. So using these tools, it's just like a paint. Okay, you have to draw it. Okay, but this is commercial. ChemDraw is commercial one. Okay, so suppose you don't have it. So there are others such as Sketchers, through which, like Marvin JS, I have given the link. So this is like the paint only. You have different options. You have to just draw them. You have to give. You can here see also SPO. So there to you can put all elements and you have to get the structure of your small molecule. So in the next tutorial, I will also show how you can do this. So okay, so from this you can draw the chemical structure of any compound. Okay, so using this you can draw, suppose this is I'm taking the case study as a amoxicillin. So this is the structure of amoxicillin and you can draw your own like this. Okay, so you can put all the nitrogens, oxygen, double bonds, rings, sulfur, everything you can put using those tools. Okay, so this will get you to the small molecule structure. Okay, but after that, what will be the formats? So I'm you know about the PDB format. This is a dot mol format of a drug. This is a SDF format of a drug. So this is actually I'm use a very small benzene. So this is the example of that. Okay, this file format are important as different software uses different such a formats. Okay, so suppose in PyMol, I guess you can open mol, but you can't open SDF or other way around. It is something like that. Okay, in few software you can open mol files, but you can't open SDF files. So these things are there. So you have to check whether which supports. But what if you have just a mol file? How you can get the PDB file or SDF file? Okay, so for that also there is this Open WebL conversion tool, which is freely available, and you can download it. Okay, so using that, here you can see the input format. Okay, here it is smiles, so you can click on this and you will see the different file formats using. So your input will be, say, mol format, so you will put the mol format over here. It will be displayed automatically in this area, and this you have to specify the output format. Okay, what is your output format? Mol2 or PDB, what format you are interested in. So you have to click that, you have to choose that format, and you have to uh, also select where you want the output file, or what is the name of output file. So you will do that, and then you'll press the convert button, and in that folder you will get to know key where are the things. Okay, before starting from this, I will always suggest you that always keep each of your folder, like each of your files in only one folder. If you are performing a docking for one enzyme and one drug, please keep it in one folder only, because that way there are many dependent files required for the software will be fetched through that folder only. So always do that. So this is a conversion tool to change the format of small molecules. It is not if you have something else and you are going to convert it, say, protein file, so that will make some errors. Okay, so always use such a kind of files. Also, they in pymol also you can open SDF file and you can convert it into PDB format. So these are the two ways you can convert the files. So this is how the first you can draw, you can get the files, and that's how you will get the .pdb file of your small molecules. Okay, so okay, so now till now what you got the small molecule structure, but it it is just the structure which you got from any repository, or you have drawn it, but you don't know whether it has a stake hindrances or not, or it's optimized it. So you need optimized to structure. Okay, so for that there are different optimization methods available. So in CameDraw you can use MM2 energy minimization technique, which is in CameDraw. There is Came 3D tool of a CameDraw. There you get this option. There is another freeware, Avogadro. So Avogadro.cc. So using this, you have it will gives you these three options. So you all of you can use. It is good to use MMFF94, or if you have a peptide, you can use the gap. Any one of them you can use, and you can perform the energy minimization. In the next tutorial, I will show how to perform the energy minimization. Okay, so this is how you perform the energy minimization, and all you have to always save in the .pdb format. So this is how you can get, and always check your .pdb file in the pymol. Always open that in the pymol and check whether the all bonds and everything are there or not. So till now we have generated the small molecule files and the like enzyme files, like protein file. Okay, so you have both structures, and by using this two structure, you will go to performing in like docking. So for the docking, you have to like prepare your ligand and enzyme. Okay, and by preparing them, you can go to grid generation docking, and then analysis. Okay, this is the overall workflow. So first start with the ligand preparation. Okay, you can see the ligand open in the Autodoc tools. So you can open from, like, it will be downloaded using MGL tools or ADT site. So I guess all of you have the links. Else you can mail me regarding that. So you can able to download the, like, open the files using file read option. So you can open the small molecule file. Okay, so few things are required. Can anyone guess ki what things we required, or if you are going to perform the docking, what things you should check for a small molecule? Let's start with the small molecules. Can anybody answer that?
Angus,
Hmm?
I mean Angus.
Okay,
Polarity of the
Angus.
side chain.
Okay, so get to know, matlab, good, but that is important for your macromolecule, which is your enzyme. I am talking about small molecules, which is a your drug molecule.
Coordinates.
Yeah. Okay, but you have the coordinates. You prepared the file, right? So you have the coordinates. So that's what just now talked about. So till this step, anyone has any problem? Like, look, I talked about small molecule structure development, and then how you can get the coordinate file or the .pdb file for that, and then the energy minimization. I will show you in the next tutorial how to do that. But have you got why we are doing it, or what is the important? If anyone has any question, ask me. Um, I guess not. But yeah, so the answer to your question, my question was, like, you have coordinates, right? Now, what you need is if you there are hydrogens present or not, or what is the charge of your small molecule, right? If you have missed some hydrogen, or what is the, like, if there is any metal present, then there might be some charge to the overall structure of your, like, this moiety. Okay, so you need to do that. You need to check that. So this is the edit. In that you will get to know to, so from this you can add hydrogen charges and set atom types. Okay, these three things you have to do. You have to first add charge. So in this case, you have to first add all the hydrogens. Okay, then you have to add charge. So I will just tell you. Okay, so you have to add hydrogens as I talked about, then add charges. So there are two types of options you will get. One is Coulomb charges, another is Gautier charges. Now you might be having question, ki what are those? These names are pretty weird, or someone, if someone is from chemistry, they might know this. But so these are the charge models prepared for each atom. Ki what will be the charge in each state, like in Mg2 plus, or what are their partial charges. Okay, so these every record is there. So Coulomb charges are for amino acids. This model is developed for this template is developed for amino acids only. So if you have a peptide or you have enzyme, then only you can use these charges. Else you have to use the Gautier charges. So if your small molecule contains a part of a peptide, okay, then only you can add the Coulomb charges. Or whenever you will add the Coulomb charges to the small molecule, the overall charges will be 0, and the Gautier charges, it will nullify the charges according to. But in ligand you have to add all hydrogens. Okay, So you have using this you can add hydrogen and charge to your small molecule. Okay. The so now what you have done is you have like stabilized the charges for your moiety. Now you have to prepare your ligand. So using the previous, this is your ligand panel. So this has a different inputs. So I will discuss each one of them. Okay. So using the ligand preparation panel, you have to prepare your ligand. Okay. So in what are the options there that I will discuss. So whenever you are going to open the ligand, there you will get one pop-up saying what are the torsions present in your autodoc like in your small molecules. So can anybody tell me why torsions are important? Because you can see in the slide also there are torsion tree, choose torsion, everything is there. So why this is important? Why torsion is important? Can anybody think of anything?
We can find out the phi and psi angles, and we can use for Ramachandran plot, I guess.
uh, but
I
phi
guess
and psi are for protein. We are not talking about protein, we are talking about ligand. Ligand means small molecule.
I guess to predict the correct conformation of the ligand, conformation of the small molecule in which it will dock in that particular.
Yeah, pretty close answer. But the important thing is let's go back to the structure.
So we can determine the flexibility of the ligand by this torsion. How much the ligand
Yeah,
is flexible to react with any kind of mod-
yeah. So now you can see the two-dimensional structure. I have shown you the two-dimensional structure, and now you can see here the three-dimensional structure, right? Now you can see this benzene ring, right? It is attached with the single bond to this carbon. Okay, so it is a possibility that this can rotate with this bond around this like around this bond it can rotate. Okay, so thus such a kind of rotations are possible, right? So your small molecules need flexibility to rotate around different bonds so that it can produce different conformations or different structures, right? What you are trying to do is you are docking your small molecule with your protein. I should have shown you, but it will be clear in the next tutorial. Key, if you have opened a small PDB file, like enzyme file in a PyMol, what do you see? You see beta sheets, you see alpha helices. Also, you can see the blank like spaces. Okay, your active site will have some kind of a space, and that space need and there your small molecule has to be go and fit. Okay, so for that, your small molecule has the flexibility, need that flexibility to fit into that. Okay, whenever you put your hand in jar, you have to sometimes little bit adjust your hand. Okay, so that adjustment also required for your like small molecule structure, and that will be given by torsions. Okay, so whenever you uh, like use this panel, you will be having these different options. So what they will see, so the first one is the detect route. So detect root is the choose smallest but largest sub tree. So what is that? Okay, okay. So suppose you have this. Okay. Okay. Suppose you have this. So what is the tree? I will tell you. Some of you might have seen binary tree. What is that? So you have this bond. There is this one end and this another end. So these are no points. These points called nodes. And this called edge. Okay. So each point is connected through this edge. So this is one tree from this point. Okay. This point you can see this tree, this tree. If you fix this point as a root, this will be another tree because this each and every point is connected with this bond. So this is called connected tree. So this point is connecting each and every residue from, so this is a path. So this and this, so this is a path of this 2 points. So these 2 points are connected with this path. Such, so root will be a such a point through which each atom is connected through some path. Okay. So that will be calculated by this particular software. You don't have to, if you know where exactly the route, you can do the manual search, but this will be done automatically. But what it is, as sir told, you have to know these things, you have to keep your mind open while you are doing the docking. So these are the points sir was talking about. So what it is doing, you you can click on it and you will get the result. That is a different thing. But by clicking on the particular point, what you are getting, that is also important. So you will detect the root, which will choose that. Then there will be choose torsion. So what it will do, it launches an interactive browser for choosing rotatable bonds. Rotatable bonds will be shown in green, non-rotatable bonds in the red, and bonds in the potential rotatable. So what this will do is, this will tell you key where you have the rotation possible. Now, why this is important? Okay. Suppose in your case, you want to fix some group. You don't want to rotate it. Because of some reason, you don't want particular like bond should not rotate. So you can do that using this command. Okay. So you have control over torsions key. Okay. This should move, but this should not move. Okay. Suppose in some case study, you got that because suppose in DNA. Okay. So DNA, you have helixes. Okay. So in that, whenever benzene planar thing goes, it will have a very less rotatable bonds. Okay. So for such a kind of thing, if literature says that this has been observed, so you can also do that in this scenario, and you can choose the torsions. So there will be set number of torsions will tell you how much torsions you need to put. Okay. So this is about the small molecule torsions. So the next thing is aromatic carbon. So what aromatic carbon will tell you is aromaticity criteria. So this, whenever you click on it, it will open a automatic window showing the 7.5 degree. So what is the 7.5 degree? So suppose in a benzene ring, you have two bonds, right? So in a benzene ring, you have total 6 carbons connected via different bonds. So if you take two adjacent bonds, so the angle between those will be the angle between the normals to adjacent angles in the cycle has to be less than 7.5 degree. So this has been observed. Okay. So I will try to check whether I'm able to open it for you so that this thing will get clear. So yeah, whenever you will open Autodoc tools, you will see such a kind of a thing. So always wait for some time so that it will open, because these will load a lots of modules, lots of libraries. So always wait for some time. So meanwhile, you can ask your questions if you have any.
Sir, I have a doubt, sir.
Yes.
Sir, you said Open Babel is for converting a mole to a PDB. Then, while we are using in ligand preparation for adding hydrogens, why are we using this Open Babel, sir?
So actually, whenever you are going to use this Autodoc tools, so this is also dependent on some tools. Okay, so for adding hydrogen, it uses OpenBabel. So OpenBabel has that like that software. Okay, and it is a free software. So this software has incorporated the OpenBabel software. So good question. Because of this, you get to know that you can it's not necessary if you are using this software, it is using that software only. It is dependent on other softwares also. Okay, so I will quickly show you. We have less time. So this is the Autodoc tool interface. Okay, you go to read molecule. Okay. Suppose you have this amoxicillin PDB. So this is how you will open it, and you will see it. Okay, so edit. So in edit command, you will have atoms, add charges. So this pop-up will come. You have to add charges. Now this window you can see whenever you click on add hydrogen. So you in the background, as told you, there are different modules and libraries. So it uses Open Babel library and add the hydrogens. Then in charges, you have this too. So as I told you, this column charges are for peptides or amino acid like structures. So if you add it, you can see the total charges added are 0, right? Because the charges for this compound are not there. So you have to compute guess share charges. So this will add charges to your molecule. Okay. Now there is another atoms, atom types. So uh, why this type is also important, because this type will tell you. So this software has different types, like one carbon is also predicted, like in some cases carbon is C1 or C2. In this way the numbering will be some cases Ca, Cb, this kind of a numbering. So each software has their own way of representing any atom or any molecule. So that will be done using that. So this is the ligand where input you will choose. You have this your because you have already opened it. So this pop-up will come. So this will tell you you have added gesture charges. The 14 non-polar hydrogens has been burned. 9 aromatic carbons are there, and torsion there are torsion degree of freedom are 7. This is TOS DOF, degree of freedom. So these are 7 places where torsion is available. So this will be shown over here. So in Go, if you choose, it will detect the route. Now see, it has detected this route, which is connecting to the every point and which is the smallest path for everything. Okay. Each bond. So torsion, it will you have detect the route, show root expansion. So it will show key root expansion where it lie. Okay, so this is choose torsion. Now you will see here. Now you can see Okay, so greens are rotatable. So you can, as I told you, so this bond, so this can rotate. So that's why this bond is rotatable. But you can't rotate the bonds between your which are in the aromatic like aromatic carbons cannot be rotated. So these are not rotatable. So that can be seen. Now you can see magenta is non-rotatable. So this bond is non-rotatable according to the Autodoc tools, like this software. But so you can suppose make this amide rotatable. So now you can see there was amide bond. So I made its rotatable, and you can make it rotatable and non-rotatable using this option. So whenever you change this, the number of rotatable bonds will be changed. Okay, so I have done. Choose make it non-rotatable, and that this kind of functions you have was I was talking about. So you have a control over this rotatable bonds, and then you, so you have choose that. Then you can go to ligand. So set number of options. This will, so you have 7 rotatable bonds, but you have to make less less rotations so that you can do from here also. So you have control over rotations. Okay, so this rotations are, now you have aromatic, 9 carbons are aromatic, so you can click on aromaticity criteria. You can choose the, enter the angle in degrees. So this is 7.5 which has been seen. So aromaticity will be checked using this. Okay, so those, this suppose this carbons, there will be normal to this one, and when the angle between normal is less than 7.5 degree, then these are called aromatic criteria. So this is, this chemistry is already they have checked, and using that only they have given this. But you have to always check that. Then you go to output, and then you save it as pdbqt. So you will say ki, till now we have a pdb. Now this tool always uses this PDBQT format. What is PDBQT? The PDB file with Q is charges and T is type, atom types I talked to you about. So PDBQT is a file which contains all these two things, and that's where the extension will be always a PDBQT. Now you save it and always save it in the same folder, your working folder. Make the folder, now mine one is in the desktop, which is this one, docking files. So I always save it in the .pdbqt format, and you will save it. Now I already have it, so I can replace it, but it's no need. So now you can do this. Now you have done, like, prepare your ligand. So you right click on it and you can delete it. Okay, so this will delete the section of this, I guess. Yeah. Okay. Now, what you want is you. So now we have converted our file into PDBQT format file. So this is how you prepare your ligand for docking. Okay, so you're like giving the charges, you're giving the atom type so that it can run in this software. So now we will go to enzyme preparation. Now you delete, as I told you, you will delete it and you will open the enzyme file. The enzyme file, you have to add hydrogen and charges, as I told you. Now whenever you will add the column charges, you will get the column charges, and just that you have to do. You have to add all hydrogens, you have to add charges, then merge non-polars, select atom types, and then you have to save it as a pdbqt file. Same thing for enzyme. So now you have prepared your like small molecule and enzyme. Okay, so this is how you will do it. Now the next thing is grid.
Excuse me.
Okay, yeah,
Yeah, what was this atom type AD4? What is meant by AD4?
AD4 is Autodoc 4. So Autodoc
Okay, okay.
tools,
Sorry, sir.
we are using the Autodoc 4.2.6. So that is 84.
Yeah. Okay.
So now you need is a grid. So can anybody tell me why this grid preparation? Yeah, you can see the grid right now. So can anybody try to think of why grid is important?
I think
I will tell you what is
limitation.
grid is.
Am I audible, sir?
Yes, yes, yes.
Sir, I think there should be a limitation of the area where ligand is going to be interacted. So I think here a grid can be exploited.
Yeah. So the grid is, you will define this cube. This is, you can say, red, green, and blue cube. So this cube will like, give you the area where you have to perform the docking. So there are two types of docking. One is called blind docking. Another is called targeted docking. Okay. So as the name suggests, blind docking means you don't know where the active site is. So I will just in a brief tell you what is the active site is. The active site is a site where you know key the residues of you, like you have an enzyme and there will be different residues. Okay, so the few of residues, through the experimental techniques, you will get to know, like there are like you do the mutations, single point mutations, and you will check the activity, and the using that you will get to know which are the active site residues. So the residues which like interact with your small molecules are active site residues. Okay, so in the literature, whenever you see your enzyme with some ligand in experimental PDB structure in RCSB, if you search, you will get such a kind of if it is solved, then you will get such a kind of structure. From there, you will get to know yeah, this particular residue is from active site. Okay, so as I told you, there are two types of docking. One is a blind docking. So in case of blind docking, you have a very new enzyme in your hand and you don't know where the active site is. So in that scenario, you need to like put all the items to the enzyme in a box. Okay, I will tell you, I will show you how it will look like. So this is a grid option through which you can open. So if you don't have open, so micromolecule, you can open your. So I have prepared enzyme PDBT prior to our module. So you can open the enzyme PDBT. So it will show this pop-up, which tell you there are partials or charges already in your. You have to keep. You want to preserve it or not? You have to keep it or not? So yes, we have to keep it. So this pop-up will always come. So this pop-up will tell you if you have some kind of error according to this tool. So what it is showing you that the charges are already added and contains no non-bounded atoms when there is no gaps or anything like that. So this is much important. You should always get this. If you are not getting this, means you have problem in your enzyme. And now you go back and check what is the problem. So this is, so in set map types, you will have this different option. Choose. You have to choose ligand. Suppose you have to perform some covalent bond so that you can do. But mainly we will go for this. Okay, sorry. So grid, set atom types, open ligand, because we have open enzyme. Now we have to open the ligand. So amoxicillin, PDBQT which I have opened. So it will show somewhere. Now grid. So you have opened enzyme and your ligand here. So grid box. This interface will show you. Now you can see this. All this box is there and all the items are there. So now using these three, so this will move your center of this cube. Okay, and this will expand. It will increase the size of your cube. Okay, this is the spacing. This is like this cube has a grid. Okay, so this grid can be optimized using. So this will also increase the space. Okay, so this will be from, it will, you have option from 0.2 to 1, and whatever he comes here is like spacing between two carbon like optimized spacing between two carbons, which is 3.4 0.347. So you can try to use that one only if it is possible, or but if it, you can always increase the space. So this is called a blind docking. Okay, so in case of a blind docking, you are trying to put your whole enzyme in the active site. Now you have to forget about this. So in the, here you can see this is your small molecule structure. You don't have to worry about this. You just have to worry about your enzyme. So now you can see all the atoms of my enzyme are in this box. Okay, so now this, if you keep this as a grid, then this is called a blind docking. Okay, because you are blind about the active site. Okay, so this is called a blind docking. Now in case this is a beta lactamase enzyme which is studied in our lab. So due to the literature and study, we know the active site of this is a serine active site. So serine residue in the STFK loop is so this is STTK loop. Sorry, yeah, STFK loop. So this is the loop. So this is my active site over here. Okay, so now I can go to this. I can show it as a, suppose. So I will show you. So now I can reduce this and I can uh, make this box around that residue and nearby residue. So there will be a more than that. So you can show it. Let me try once. So okay. So these are the different options to show. I forgot to tell you to like to change the representation of your enzyme. So now you can see it, and you can hide the, yeah, now you can see, and here you can see the serine residue. Okay, so if I make the changes and if I uh, make my center around here in this area, if I move this like this, so yeah, so if I move like this and if I come around this, if I make the box around here, so this will call the targeted docking, and I can make this. If I make the grid around this point, so I'm not, please note that I'm not right now making because it will take our time, and I don't know till how much time my backup will work. So file, you have to close saving current. So this will save your grid. Okay, so this is how you set your grid. So the in the targeted docking, you will be having specific list of residues. Around that residues only you can make the, you have to make the grid. And always keep in the mind, whatever you do here, please make, note the centers, all the XYZ centers and the number of points, all this thing and the spacing, everything you have to note down, because whenever you are going to write your in thesis or in your research article, you need these points. So always make sure you have noted down everything, which the results you are getting, but the setup you are using. So I talked about everything. So there is one file ad4.bound.dat. So that is called a parameter file. You have to keep this parameter file always. And another thing is that whenever you are going to save this, okay, so yeah, so whenever you are going to save this, so now you have make the grid, so you will output and you will save as a GPF file. So after saving this this you have to save this in the same directory first of all, and you have to give this exact name, grid.gpf, with the extension you have to write over here, and then you have to save. Okay, always make sure that you do this. If you just write grid, then it will save as a grid, and .gpf for like extension will not be there. So this kind of things you have to keep in your mind. Okay, so next thing is docking. So you have made the grid. You now this is key which kind of a tool you are using. Like there are different algorithms for performing docking. So you have to set them. So this will be using this panel. There are all the options you can see. First is a macromolecule to set the macromolecule. Second is ligand to set the ligand. Another is search parameters, which will search your parameters, the docking parameters. So I will talk about them. So I have enlisted them. So in docking algorithms, you have this 4 options, or in parameters, you have first 3 options. So what are those? First is genetic algorithm. So I will not go into detail, but I will give you an idea is that a population of a trial confirmation is created for this, and then in a successive generation these individuals mutate, exchange conformational parameters, and compete in a manner analogous to biological evolution, and ultimately selecting individual with the lowest binding energy. Means what it is doing, it is making one conformation, okay, then mutate, it means it's rotate or translate its position, conformation. It's not actually performing the mutation. The mutation, it means it is changing its orientation, it's mutating its position. Okay, it's making bond rotations and all. And in that manner, it will check key what is the, like in the particular site when you are going to perform the docking, how many, like how this conformations are changing, and what is the lowest binding energy. So it will calculate the energy, and it will tell you what is the lowest energy one. So another is simulated annealing. Simulated like performs a more global early search in the run, when higher temperatures allow transition over energy barrier, separating energy valleys, and later on performing more local search when lower temperature place more focus on local optimization in current value. So simulated annealing is like it is over temperature. So it when you have a higher temperature, the bonds will rotate more, and according to that it shift to the lower temperatures and check the optimization, the energy. It will also check the energy of your ligand, and it will check whether the uh, what is the optimized position. In local search, only the local space is sampled, performing a sort of minimization of a ligand in a current position. So it is a minimization of that particular ligand in the grid, whichever you are going to set. Okay, so and another is Lamarckian genetic algorithm. So Lamarckian aspect is added. So it is actually a genetic algorithm. Is just that it allows individual confirmation to search for their local confirmation space, finding the local minimum, and then pass this information to later generation. Okay, so it will always change for the like or like suppose it is rotating only one bond, so it will show in that space, it will find the minimum one and use that one to the next rotatable bond. Suppose you have a 5 rotatable bonds. Okay, so one it will rotate, it will find the lowest one, then it will that confirmation, it will take the second one, like that it will go. So for this you don't have uh, uh, you might have not got this, but it's fine if you don't get it, because this algorithms has been verified and uh, s- it has been seen that Lamarckian genetic algorithm is the best algorithm for your most of the rotatable bonds, and this is the latest algorithm. Okay, so that's why you will use the Lamarckian genetic algorithm for your this and you for your docking, and it is based on genetic algorithm, so all the parameters of genetic algorithm will be applied to it. Okay, so y- suppose you are using Lamarckian genetic algorithm, so for that all the search parameters will be for genetical will be exported from genetic algorithm. So when you click on that, you will choose the genetical algorithm which has the parameters of Lamarckian GA. So there will be number of GA runs. I mean, how many like total confirmations you want. Suppose we have keep it 10, you can keep it to 20, 25. But since the number increases, the more the like process will happen. So maximum number of evaluation will be made and recommended for highest binding. You may use long, but it will take more time. So this is what I was talking about. So suppose you have a torsions from 1 to 10, then you have to keep the evaluation to the medium. मतलब the iterations it is going to check for each evaluation has to be this much. And if it is greater than 10, then you have to choose the long one. For this, you need a good system also, and keep everything is as default. And all the docking parameters you have to keep all defaults, another, and you have to output, you have to choose Lamarckian. I will show you about what I was talking about right now. So in the docking, you will choose the macromolecule, set rigid file name. Okay. So your macromolecule, we will always keep it as rigid. If you want a flexible one, so then you have to choose what are the flexible residues and all, but which is in the special cases only you need that. So we will use the enzyme, and always make sure you use the file name correctly. So in case of macromolecule, if you use the ligand, then it will going to hamper your docking. Then in ligand you will choose amoxicillin. Now it will show in case of your small molecules, what are the atom types. So this was I was talking about. So these atom types of are this one, center of ligand, the root is this one, and number of active torsion angles are 7. So you accept it because you have already seen it. So that's what I was talking about. In search parameter, you have this 3 algorithms. So you will choose genetic algorithm. So it will pop up this one. So this is so you can always make this to 20, 25. So the number of evaluation is this. So if you choose the short, it will be less. If you choose medium, this will be, I guess, 25 lakhs. So you have to keep the maximum generations has to be 27,000 only. So this will be parameters which are useful for genetic. If you want the details of each parameter, so there is a manual, as I told you in previous class also. There is a manual of this Autodoc tools where each and every, like how much you can change, that is specified over there. But these are the optimized values, so you can always use them. So uh, so this is then it will come to docking parameters. So there are different docking parameters it uses. Like what is the parameters for running the algorithm, okay, which contains random number generation. It will contain energy parameters. So you can always go and check what are the parameters. So external grid energy, maximum allowed initial energy, maximum number of retries, how much time it should retry. So everything you can see here, but these are very optimized parameters. So this is like, so know this, keep whatever you are doing here, it is going through iterations, right? It will check one confirmation, then it will check another confirmation. So these iterations will be there. So in each iteration, what should be the translation? So all these things will be related to that only. Okay, so these are the options. So keep it default because they have optimized it. So you have to like check the literature. From literature, if they have specified something for your enzyme, then only you can change. So other options you don't have to do this, but yeah, you can try to check it. It's accepted. And then you have to output. In output you have this algorithm. So you are, which algorithm you are going to choose is specified over here. So you use this Lamarck-Ng algorithm, and it will pop up, give you pop up, and you have to save it in dog.dpf file. So you choose this, you write this, and then you save it. So for me it is already there, so I will not do that. So this is how you will do the perform your docking preparation. So till now you have got, like, you made the small molecule and PDB structures, then you got your grid, then you prepared your system for docking. So this is for docking. And Lamarckian genetic algorithm provides you the most efficient search for general applications. So this will give you to the point where you have to perform this. So these are the two commands which you have to run. And so there is a option in here. So there is option, there's run autogrid and run autodoc. But sometimes due to some setups, you will not able to, like, this will fail. Okay. So its better way is to do them using command prompt or Linux terminal. So I will show you for the Linux terminal, you have to go to your terminal and you have to open. So those who are like habituated with the Linux, you can you can do that. So for sorry, but for Windows user, I will show you. So you go to start menu, you write cmd or command prompt. So this will open. So this is a command prompt. But another thing you need is, where are your docking files? Okay, so suppose this is my folder. So in Windows, when you click on this bar, this is called the address bar of Windows. So this will give you address. So which is saying it is in the C drive, user, my folder, desktop, and tutorials and desktop. This is a path of that my folder. Then you copy this path. Okay, then you minimize it, and you go here and you write cd is change directory space, and you paste it over here. Okay, so you can see the whole path is, or you can write it, write this also, and then you press that. Now you can see the blinking cursor is after this, means you are reached to this point. You need to exactly see like this one only. Okay, and suppose in Windows, now I'm in the C, but you have to change the drive. Okay, so D, suppose I have to go to the D drive. So this D, then colon, then it will shift me to the D drive, and then you can paste your D drive. If I'm in the, now I'm in the C drive. So what I will do, I will go back to the C, and then it will automatically give me there. So now from this you have to write this auto grid 4, then space minus p space grid dot g gpf space minus l. It is not 1, it is l minus l grid dot glg. Okay, let me see if I can show you. So what I will do is I will try to delete this file. Okay, now you see. Okay, now keep eye on the folder. Okay, so I have written this command as you see. Now you press the enter and you will see what is the output. Or now you can see, sorry, I can't find grid.gpg because my file was gpf. So this you have to make sure you write it as I have written. So grid.gpf minus l grid.jln, you put the enter. Now this cursor will blink and you can see these new files which I have deleted previously were prepared. So you make sure you wait until you see the address bar again, because this is running in the background. It is happening. So you have to wait for some time unless you find this blinking cursor after the address bar. So you'll wait for some time. Now you can see this address, like this address is came and the blinking star is after that. So this means this command has run. Okay, so after this you will perform the doc command. So sorry, this is L, but it should be K, but it's fine. This will also work. So doc.dlg. So autodoc 4 minus p doc.dfib. It has to be same like this. Minus L doll or doc, whatever. This name you can change anything, but it has to be .dlg. So this is very important. The extension and everything are play important role in this. So you have to open this and this will run your autodoc, and after that you have to wait for some time because this will take sometimes 15 minutes, 30 minutes, or 1 hour or so. Okay, so this will take this much time and you will perform the docking using this, and you will get the .dlg file. Okay, doc.dlg or whatever the name is, name.dlg file. Okay, so which I can show you. So this is the file. You can open it in the notepad, or the software kit. So using any such a software, you can open this thing. Now this one contains each and every, like you have to choose the 10 confirmations. So all the details about the confirmations can be seen in this. So everything, so all those confirmation and everything. So this is actually a huge file um, containing details of each and every output prepared, used, all the parameters used and the outputs has been written over here, but you don't need. So you can see the like atoms and the coordinates over here. So it has prepared all the 10 confirmations. So you search, put the control F and you search for histogram. If you search for histogram, it will show you the histogram. So what is this? So first one is now I guess you can see very clearly. So cluster rank. So what it does, it's as I told you, it try to find different conformations. Okay. So on the basis of energy, it like make the cluster of nearby energies or nearby conformations, and using that it has made three different clusters. So cluster where, but in each cluster you can see in the first cluster there is only one, in the second cluster there is also one, in third cluster there are eight, which you can see over here. Okay, so on the basis this are performed, and you can see at the third run is the lowest bending energy run, so which is minus 6.29 in this case. So the more this minus 9 or minus 8 or minus 10, the your like docking is that much good, the binding energy is good, but this is intramolecular binding energy, I mean between the atoms. Okay, so the third confirmation from your this is the lowest one, so minus 6.29 the energy for this cluster is. And you can see here the RMSD table, which is showing the binding energy. So first, this is the first rank cluster, which obtained at the third run. So we have run for 10 confirmations. So third confirmation out of the 10, out of this 10, it has the lowest binding energy. The another cluster was this one, which is at the seventh one. So it has the both the cluster has only one. So this one, and these are the rest eight, which comes under this third cluster, which has the mean binding energy minus 5.27, and the lowest is this, you can see minus 5.48. These are the rest of the eight. So since you, according to your choice, like if you want, if you know the confirmation, you can choose for that confirmation, or you can choose the lowest binding energy confirmation. So this analysis, I will not talk this because it is going to take a time. So this is how you can choose your structure, like what is your final structure, and you make the complex. Yeah. So in the docking analysis open, and you can see the all your results. So by using play confirmation you can play the results. So I will in details I will show you. So you have to convert them, so you get the results, you get the DLG files. Let me see if I can show you that. So I have 40% is still available, so let me try to show you. Okay, deleted everything. Analysis, docking, open. So that output file is doc.dlg file, as you can see. Analyze macromolecule. You have to open your enzyme.pdbqt. Okay, so this is your enzyme pdbqt. Analyze, confirmation, play. So there are two options: play by rank. Play will play from 1 to 3, like confirmations, first one, first output, second output, and energy. Then it will as a cluster. There is third one first, like the order which we have seen in the our RMSD plot one. Okay, so this, okay, it is not showing because, yeah. Now you can see here is my enzyme, okay, and here is my output, as you guys can see. So now if I go to next, now you can see it is changing the different conformations. So you can play it. So now you can see all the 10 conformations. First, second, third, fourth, fifth, sixth, seven, eight, nine, 10. All the 10 conformation you can see. Now you wish to save your all the results. Then you first set your directory. So there is in the preference, you set your directory. You paste that path over here. Okay. So that directory you have to check the path and you have to paste it over here, and then you have to click on the set. So that this was my path. Then I will paste it over here. Don't know if it will paste. Yeah, pasted. Then I will say set. So this directory it's set. Okay, I have to delete this. Now you go here. You click on this button which says open panels to change and play. Now this will come. Now using this, you have to write all, which will write all confirmations over here. Okay. So you have all the confirmations written over here. Now you can open them in the PyMol. So these are the PDBQT format files. So you have to open them in PyMol and then you can save it in the PDB thereafter. Okay, so this is already I have given in my presentation too. So here you have to write this. You have to, if you want to save, you can save the each one only. So these are the in those who are using Linux. Those who are using Linux, they have other option. They can use this commands and they can directly convert their DLG file into the PDBQT and thereafter PDBQT to PDB as well. So they can use that. But those who are using Windows, please don't use them because these softwares are not installed in your like Windows system, so you can't use them. But yeah, as I told you, from using the options you can save them, and from thereafter you can open into the PyMol and save as the complex. Okay, so you have the output runs. Then now what remains is analyze the complex. The tutorial, we all will analyze this complex and we will check how we can do that. So there are like using PyMol also you can do that, using MAT, which is our tool. That is also you can do that in using PLIP server, so you can do that. So this is output of this PLIP server. So which is showing this is the ligand and these are the different residues which has connected with the hydrogen bond or hydrophobic interaction. So this is very important step you have to always look for, is that you have to find all the interactions. You have to make this, and this you from this you will get to know ki which amino acid is interacting with which like atom of the small molecules, and what is the distance, if it is a hydrogen donor, sorry, hydrogen bond donor or acceptor. All these things you will get using these two server, and that is how you will like get ki what kind of interaction you are having, and then you have to note this down. Okay, key 92A residue is interacting with this O2 atoms of my ligand. Okay, so this you have to write it down and then you make your own figure in PyMol. This is a figure. So this is how you make your figure, and you click on you so that's now you will get to know why PyMol tutorial was important. Because this you can generate using the PyMol tutorial, the first tutorial which we had. So using that only you can do this thing, and then you can make your own table. You can make your this own interaction table using the residue name, atom name, residue name. So this is one example. So this is, you can do that, and this will tell you ki what are the interactions are important in here. So you can use our mat tool for that, and using that also you can get the active site, protein ligand interaction, everything. So there's tables also, you can actually get it from there. But after so this will, so everything which I talked about will, what it will tell you ki you will get the results. But now after getting analysis from any web server or anything, what you need is, you need to check whether this is correct or not, because their softwares will have some limitations. Their software, all the in Autodoc, you have to check. So from that checking you can do using the Pymol. You can open this into the Pymol, and all you can always make, or you can always check like this, ki what is the distance. So what is the distance, which atom is interacting with which atom. So that you have to see, and that you have to make. This will get you all the insights about your enzyme and uh, your small molecules. So this kind of interaction can be analyzed using the Autodoc tools. So apart from Autodoc tools, there are other docking softwares available, but they are commercial. Like from Schrodinger, you have, from Discovery Studio also, you have different softwares present, but those are commercial. So this is the free software you can use. Apart from that also, other free softwares or other online web servers are there. But in this Autodoc tools, you have, like, you have control over your, what you are doing, and using that you'll get such a kind of results, and which will help you. Okay, so which so this was the serine I was talking about. So this serine you can see interacting with my this moiety, my small molecule moiety. So this is showing me ki, yeah, the results are coming as expected. So this also you have to check ki what you are expecting, you are getting or not. So this is much more important. Okay, so do you guys have any questions you want to ask? So I will just have given you overview. I will just summarize once what I have done. So first we gone through small molecule, how we can prepare the small molecule. In last class, we have talked about protein or enzyme. So the structure formation using homology modeling we talked in the previous class. And there, from there, using that model, you can go to, you can matlab, that is useful in our docking. So we prepared a small molecule. So we have structure of enzyme and a small molecule. So using this two, we perform, like, we prepare the ligand, we prepare the enzyme for docking. Then we set the grid, we set docking parameters, then we perform the docking, and then we analyze the docking. So this kind of overall workflow you have seen, but due to the present circumstances, we haven't done it. Like, we haven't done using the practical session, but that practical session will be organized in the next Sunday, and there you will get more ideas. But till now I have shown you kind of a steps, so you go through, so that why you should grow through. And it is, I will say, this happened, whatever happened is a good thing, because if I tell you to do these things, and you are doing it, sometimes you will not get the problems, but when you do on your own, you will get the problems. So I will suggest you to suggest all of you to go through my tutorial and perform these steps, and then you will get to know ki where you got stuck. And then only, whenever we are performing in the next week, then you will get to know ki, ha, this is where I was stuck, this was the problem, and also you can discuss your problems with me. and I will ask all of you guys ki what are your problems where. So that will show me ki you have done or not, because this is not so easy task. Once you do, then it is easy, but for the first time it will give you so many problems. So I want to know ki what are your problems and what you came across. So I want to know that in our next tutorial. So I suggest you to go through all the all these steps and everything, this PDF. If you don't have it, this presentation, I will send you. So ping me. So there is no option over here, but yeah, I will all or I will send you the PDF file as soon as possible. Uh, I will share the one drive link. So using that, you can download all this enzyme file or small molecule file, which is energy minimized. But you have to make it. So I will tell the name of that. So name is there, amoxicillin, in the file name. So enzyme file, amoxicillin file, the one 84 file is required. That I will give you, and using that you have to you have to make sure you are performing it. And I will send the drive link to you. Check that drive link after from tomorrow onwards, because today I will not able to upload it there. So I will make changes in here so that you all will get to know ki what where the steps are. So you perform the steps and let me know ki what are the problems you came across in the next tutorial. So if you have any other questions right now, please do ask.
Yes, I was having a question. So, when we are downloading the small molecule structure, either in .mol format or .sdf format, so as you have told that .mol format, before converting it to .pdb also, we can view it in PyMol if we want, but we can't do that in case of .sdf.
I'm not. I guess you can open SDF format in PyMol, not Mol. For that, there
Okay,
are
okay.
other software
The other way
that
around.
like Rasmol is there, from which you can view. Rasmol is there, yeah. From Rasmol, you can view Mol or SDF both.
Okay, and when we are converting .sdf to .pdb, so in the Open Babel tool, so I just was doing it, trying to do it, and I saw that in the input section. So how do we put the molecule there? I mean, I was not able to do that. I was able to select the input format or the output format, generate coordinates 3D, that's all right. But how do we put the molecule there? Do we copy it or what do we do? I mean, I just got stuck in the converting step.
So this is a graphical user input. So GUI is graphical user input interface. So here you can see, here you have to choose. So say it's suppose I have a mol2. Okay, mol2. So it will all load everything here. So you click on this. You click on this, uncheck this input below. You uncheck this. Then you click on this 3 points, which will tell you key from where you have to get. You go to your folder and you choose. I'm already in that folder. So we'll choose and I will open. So this is how you open and it will get. So you have to uncheck this. Then you have to convert it into the SDF. So you go, you choose where you have to put it. So I want to keep in docking files. So I will save this so that you will see both things. Okay, you have to provide the file name. Say I will give the different name, test.sdf, or you have to choose whatever the format you want to convert, and you have to give that name only. Then you save. So it will output file will be in the, so this path, both the path you will see over here, and then you will press convert. And then it will take time, and then it will convert it over. Here also you can copy paste from here to notepad, or you can go and check over here. Now I guess I should have that test.sdf file in my folder. Yeah, it's over here. And test sdf can be opened through PyMall as well. So yeah, you can get through here. Yeah.
Okay, thank you.
Any other questions?
Hello.
Yes.
So actually, I have question on setting the number of torsions.
Okay.
So as you said, like
Mm.
as in our example, there were 7 number of torsions, right? So if we uh, minimize that number of torsion,
Mm.
so let's say to 5 or 4. So will
Mm.
that will that Autodoc make rotatable bonds into non-rotatable bonds on its own, or do we need to specify that bonds?
So that will matlab, it will be chosen randomly first of all, because you're not providing which bond to like stop. Okay. And only the torsions you have specified, suppose from 7 to 5, so only 5 bonds will be rotated at the same time.
Yes.
Yeah, so it
Yes.
will be
So
on
we
the random
cannot
basis.
specify that.
We cannot specify at that point, but before that I have shown you an interface, this one. Let me,
Sorry.
so my power will, so in here you have this torsion tree, you have this option, choose torsion, and this will open this thing. So here you can make the non-rotatable and rotatable, this kind of thing you can perform here. So that will be shown here ki how many numbers will be there. Okay, so that you can perform through this panel.
Oh,
Yeah.
thank you.
Yeah, so I will go through chat once. uh, so can you please tell me when same way you have uh, got the date and time for this class, you will get this similarly, but it will be on the uh, next Sunday on the same time. While I'm trying to add H, so this is from Upasna Hazarika. While I'm trying to add H in ligand preparation and prepare the output ligand.pdbt, a problem arises. There is 6 hydrogen atoms is missing every time, and the message displayed is merge 6 non-polar. Yeah. So what it does is, when you add all the hydrogen, it will add the polar and non-polar hydrogen both. But whenever you go for input preparation, this will merge the non-polar hydrogen atoms, because non-polar hydrogen atoms uh, not that much required in your this in your study. Plus, non-polar hydrogen atoms make the calculation more like, since you have more number of atoms, the calculation behind what is going on, you are clicking it, but the many calculations are happening behind. So whenever you are performing the docking, the final step, at that time many calculations will be there, will be happening. So for each atom it will calculate the energy and everything. So from there you can get the for that you need lesser amount, and non-polar hydrogens has been seen they have not much role. So that's why the software automatically do that. You don't have to do that. It will automatically do that. So that is a default thing which is in the Autodoc tools, and which is commonly seen. So you don't have to worry. It displayed this thing. So you have to just note it down or keep it in mind and let it be, and go for a next step. So yeah, but this pop-up will come, and it is not a warning. It is okay. You should get this pop-up. Yeah. So do we? Yeah. Yeah. Do we have any other question? Let me check. Okay. Yeah, Devalina question. Yeah, recordings will be shared with you. Uh, the question from Aparna is that, can you please tell me how I will get PDF files of all 10 modules? So in the last I told you you will convert into the PDBQT files using the using that panel I talked about. So to all of you, wait. So to all of you, I'm sharing one link, drive link. So this folder, please check after tomorrow. So the day after tomorrow, from day after tomorrow, you can check this and you will get the presentation mm, and the files required here. Please go through them and then you can try. So yeah. So the answer to your question, Aparna, is that in Autodoc tools, whenever you are going to analyze your docking, there you will get that option to write all DLG to PDBQT. Now you open that, you can open that PDB, open those PDBQT files in Pymon, and then you can manually convert each and every one into the PDB. Smriti Gaur has questions. So first question is, once we have created a ligand on the software, it means we have created the molecules with all required atoms. So why we are add hydrogens in extra? Will it not change overall form of the ligand structure? Okay. If you when you are going to open this whenever you are preparing your ligand from any software, sometimes some of the softwares will not give you hydrogen added. Okay, so the for uniformity, the adding hydrogen is important, and for before adding the charges you should have all hydrogen present. So that's why we perform this step. Whether you have it or not, we perform these steps because sometimes there are only polar hydrogens added. So for that, for that you have to add all the hydrogens, and hydrogens are much important for your calculations. So that's why it is adding hydrogen is necessary step. If you miss it, then you will get the results, but that results will have some flaws. Like when the interaction, when it is going to calculate the energy, binding energy, so there is hydrogen bond energy term is there, so that will be affected if you haven't add the hydrogens. When we make a grid, the second question of her was, when we make a grid, is it not important that we take the entire molecule in the grid, because we might also have some allosteric interaction that one might want to consider. Okay, so as I told you, there are two types of docking possible: blind and targeted. In the blind docking, you don't have anything, so you always do the you take all the atoms. But in case of targeted docking, so you know key this atom, so this area is an active site area. Now talking about other interactions, whatever we are doing is we are trying to predict. First of all, we are not trying to perform the experiment, we are trying to perform the theoretical experiment where we are trying to predict. So now the predictions goes well if you have less number of calculations, and it has been and whatever we are doing is that we are keeping our molecule rigid. So that's why the effect of all other molecules atoms will not be calculated, and that will, it has been seen that the active site residues plays more important role than others. So that's why those atoms in that area, or the important loops, that is important. Now to answer to your question is that you can do that, but for that you need to perform simulation. So for dynamicity, whether the any loop is affecting or not, for that there are other methods, simulations or NMD. So using that normal mode analysis anyway, so that using those kind of things you can check that. In this, in the docking, you cannot do that. So that's why this is the molecular docking is on the molecular interaction of the atoms, and that's why we set the grid to only that particular, because the softwares are designed like that. You also get should know why we are doing this, because the softwares has also some limitations, and for that only we have performed that. Third question, can you please suggest why do we need to know the root? You don't need to know the root because it's more graph theory point of thing, but it is better if you know the root, so that whenever you are going to perform your analysis, you will know which atom was the root and around which all the calculations has been made or the rotations are happening. Okay, so from the root point of view also you can see what the changes are happening. So that's why if it is good. Okay, the link I have shared here is the drive link where you get the file, and this for each session we perform the different link.
No, no, she is talking about the mailing list. Actually I did that, but that was not updated. I did that now.
Okay,
Go to
okay,
next.
okay.
Go to next question. Yeah.
So question from Pratik: if there is too much flexibility in a small molecule, then is it possible to dock against protein structure? Yes, you can do that, but there is one thing which also I have mentioned in my presentation. If you have a molecule with a lot of torsions, you mean to flexibility means torsions. So the number of torsions will be greater than 10. If you have such a kind of a condition, as I told before, you have to run for longer iterations. So the iterations in the genetical algorithm has to be greater than that 27 lakh, more than that. So it has to be that evaluation pattern has to be long. Then you, and for that also you need a good system so that it can run that thing. Or if you sometimes in your laptop it may take a day or something, or a longer time it sometime it takes. So yeah, you can do that. Tuhin Sarkar has the same question regarding the mail. Yeah, sir will update it. And yeah, as sir told, if you have any academic question, then write over here. If you have such a kind of a questions, you can mail us separately so that we can discuss them. We can do those things. Yeah. Any other questions you have? So in the next class, sir will, as sir told, sir will talk about things in details, and we will perform the practical session where we can set and I will answer all of your question, and I will show you how the docking is doing. So before next class, I will suggest you, all of you, to check the like perform, like check the presentation. I will send it again. I will make some changes so that it will be more like more convenient, because while making this presentation I was thinking so that you guys can see it in the practical sense, but now I will add more screenshot to it so that you can guys follow each and every step, and follow that so that we will get to know ki, yeah, if you have done it or not. If you have questions, then we will know ki, yeah, you have done it. So perform that, try to perform on your own, because we have seen that ki, whenever we have giving a practical session, everybody performs that, but whenever they are doing on their own, they will always get some problems. So make sure you do that so that you will have a clear idea.
Yes, yes, yes. And to add to that, I would say that once you do the one where the files are provided from us, you go and try to like pick up a new project, deal with making the files, because once you like would be able to make your own file and perform docking, then you are like going to be independent, and then you will get a lots of questions and we would be happy to answer them. Then you are actually pushing yourself towards working independently on those techniques. And as like I talked initially, please go and try to understand the algorithm. you know we have this, like your generation, and like we who are actually coming from 90s, who have seen like no internet and then see the bloom of internet. One difference I could tell you, which is like a lot of things are good, but one bad thing is we depend too much on the internet information. It is not that something which is written in the internet is correct. So what I mean, you try to go to the best software. Like here we are talking about Autodoc. Autodoc is a tool which is coming from Scripps. Scripps is a very good institute, but more importantly, Scripps have given the opportunity to make it a open source. So a lot of scientists who are not a part of Scripps like us, we contribute with our small improvement of tools to the platform. When I say we, I mean the whole world. So that's why if you look at the percentage of you know people performing or people using tool, you will see that Autodoc and Autodoc Vina, which are two different parts from Scripps, they are used more than 80 to 85%. So you could depend on that. You could go there, you start learning. It is definitely important how it is happening. Like the tool, the magic box. But as a researcher, you should not consider that as a magic box, because then basically you become a technician, which we all don't want to. We want to become a scientist, a like a researcher who understand what is happening.
You are not audible, sir.
Yeah, I guess sir will rejoin. Just wait for sometimes. Sometime. Hello. Hello, everyone. Due to some technical issues at Sir's place, he is not able to join, and also my power backup is about to finish. So yeah, you guys mail us, or you can contact me through my number if you have. So you guys just contact me or Sir through mail, and we can discuss your problem. So thank you, and we will finish this right now, and we will meet in the next meeting. Before I go, I will share my email ID and my phone number so that you guys can send us query. Yeah, shared my contact details. Please contact me if you have any doubts. See you in the next meeting. Thank you.
Thank you so much, sir. Thank you so much, sir.
Thank you.
Thank you.